So whatever the encoded information in the gene, that is the DNA, finally expressed by synthesizing proper protein. So information to functional protein, a journey, nothing but the gene expression. All the cells are the same DNA. Why does it differ? One gene for neuron, other gene for muscle. That is nothing but gene expression. Gene expression in case of the prokaryote is very simple. Start or stop the transcription level. But gene expression in case of eukaryote is the complex. Occurs, that is exerted at various levels. Transcriptional level, processing level, transport level, translation level. Transcriptional level, this is the first checkpoint, molecular checkpoint. The accessibility of RNA polymerase in a given promoter site regulated by the accessory protein, which affect RNA polymerase in detecting the start site, that is structural gene. It has to pass through that checkpoint. Second, after formation of the RNA, mRNA, that is heterogeneous RNA, it should undergo processing, that is splicing, capping and tailing. So it should be the processing, that is alternate splicing, is time specific, tissue specific, species specific. Right protein has to produce means it should undergo in a proper way, that is alternate splicing. It has to pass through the second checkpoint. Third, transport from nucleus to cytoplasm. So properly processed RNA only leaves the nucleus, enter into cytoplasm with a cap at 5 end and poly A tail at third end. Then only it leaves the nucleus and enter into cytoplasm. Translation level, the fourth one. The formation of the protein is not going to be a complete process. Post-translational modification occur depends upon the requirement in case of the target sites. That is post-translational modification of the protein. Next, the gene it meant for one specific function or set of functions. If we take the E. coli, Escherichia coli, it synthesizes beta-galactosidase enzyme bGAL, which catalyzes the hydrolysis of the lactose into glucose and galactose. Lactose is a disaccharide that hydrolyzed into glucose and galactose.